Hi, so in cell respiration part one, we talked about the three phases and went through in detail glycolysis. At the end of glycolysis, you end up with 2-pyruvate. And if you're going to do complete cell respiration, those 2-pyruvate then need to enter into the mitochondria to go through the citric acid cycle and the electron transport chain. So that's what we're going to talk about now. So again, the mitochondria has the inner membrane, the outer membrane, the matrix. And so the second two phases of cell respiration, citric acid cycle and electron transport, happen inside of the mitochondria. So the first little technical problem we have is that the pyruvate that we ended up with at the end of glycolysis, where we had 2-pyruvate, needs to get into the mitochondria. And in order to get into the mitochondria, it's converted to a molecule that can get in called acetyl-CoA. So pyruvate, remember, has 3 carbons. Acetyl-CoA has only 2 carbons. And so as another product of this reaction are carbon dioxide. So remember, in cell respiration, you produce 6 carbon dioxide as you break down the glucose. So this is the first 2 of those carbon dioxide are made in converting the pyruvate to a molecule called acetyl-CoA. This also produces 2 of those electron acceptor molecules. So they're carrying the high energy electrons that were released when we removed this carbon from pyruvate. So the acetyl-CoA is the form that actually is used for the citric acid cycle. So we made 2 carbon dioxides. We need 4 more carbon dioxides, and those are going to be made in the citric acid cycle. So the citric acid cycle, as I mentioned earlier, is also called the Krebs cycle. It occurs in the mitochondria. It's actually in the matrix of the mitochondria, which is the very innermost part. Oops, I thought I switched that too. And it produces more of those electron acceptors, and it makes a little bit of ATP, 2 more molecules of ATP. Remember glycolysis, we made 2 ATPs. We're going to make 2 more in the citric acid cycle. But what's important here is the number of the NADH and FADH, which are the electron acceptors carrying those high energy electrons. We made some of those in glycolysis, made some in converting pyruvate to acetyl-CoA, and then we're going to make more in the citric acid cycle. As we go through the citric acid cycle, we make 4 more CO2s. So that gives us all of our CO2s. We make 2 more ATPs, as I mentioned, and then NADHs and FADHs. The carbon dioxide that you're all exhaling right now, so every time you breathe out, you're getting rid of those carbon dioxides that you made as you broke down glucose inside of your cells. So that's the source of those carbon dioxide. So if you think about it, excuse me, you start with glucose, which is C6H12O6. So glucose has 6 carbons. And when we break it down, we exhale 6 CO2s. So all of the carbon from glucose is what you exhale in the form of CO2. So at the end of the citric acid cycle, there is no more glucose. It's all been broken down into carbon dioxide, and then you have exhaled that in your respiratory system. Excuse me. Again, this occurs in lots of little tiny steps. I'm not going to go through each of those little tiny steps, but there's 1, 2, 3, 4, 5 steps in the citric acid cycle. Each of them has an enzyme, so there's 5 more enzymes needed to take the acetyl-CoA through the citric acid cycle. But you can see as you go through the cycle that here's where you make your ATP. And remember, you had 1 glucose went to 2 pyruvate. So that means this cycle happens twice for every glucose that you're breaking down. So you only see 1 ATP coming off here, but since there's 2 of them, you're getting 2 ATPs. And you're making NADH at several spots, and FADH, the other electron carrier, also. And then here are all those CO2s. Here's the CO2. Where are the other CO2s? Here's the CO2. So there's 2, but remember you're doing it twice, so that's the 4 CO2s. So that's the process of the citric acid cycle. If you ever take biochemistry, you have to know each one of those steps and what happens, but we don't for this class. So with the citric acid cycle, you start with the 2 acetyl-CoAs, which were made from your pyruvate. Oops, that's too dark. You can't really see the... and you convert those to carbon dioxide. You make NADH, FADH, and 2 ATPs. So you're making more energy, but we'll see that these guys are used in the electron transport chain. So you make 2 ATP in glycolysis, 2 ATP in the citric acid cycle. So far you've only made 4 ATPs. That's really not too many ATPs to keep your cells alive. So after the electron transport chain, glucose is essentially gone, but what happens in the electron transport chain is all of those electron carriers, the NADH, FADH, that you made in glycolysis and the citric acid cycle, are now the energy from those electrons is going to be used to make ATP. So that's why it's called the electron transport chain, because it's taking those high energy electrons, which are being held by the NADH, FADH, it removes them and uses that energy to make ATP. This is the part where you need oxygen. So right now we're all breathing, and the reason you're breathing, the primary reason you're breathing, is that the electron transport chain in the mitochondria of all your cells needs oxygen to make these ATPs. And so if you don't have oxygen, your mitochondria can't make enough ATPs to keep your cells alive, and that's why oxygen deprivation is fatal, is because you can't make enough ATPs to keep yourself alive without oxygen. The electron transport chain, I think, is the most complicated to understand of the 3 phases of glycolysis. And it's taking those high energy electrons from the NADH and FADH to make ATPs. You don't need to know these numbers, but each one from NADH makes 3 ATPs, each one from FADH makes 2 ATPs. And then you remove these hydrogens when you release those high energy electrons, and those hydrogens combine with the oxygen you're breathing to make water. And that's why you need the oxygen, is to combine with those hydrogens to make water. I'm going to show you more pictures here. So again, this takes place on the inner mitochondrial membrane. That's the one that has all the folds in it. And what's coming in are the NADHs and the FADHs that you produced in glycolysis and the citric acid cycle. So those have those high energy electrons. The energy from those electrons is used in an active transport system. Remember, active transport is where something pumps something across a membrane against the concentration gradient. And the energy is pumping hydrogen ions outside to the space between the two membranes. So you have all this hydrogen accumulating here. There's a bunch of membrane proteins that do this. You don't need to worry about that part. And so what happens is that you get a buildup of hydrogen ions in the space between the two membranes. So there's a whole bunch of them there because they're all being pumped out. And then this final membrane protein here is actually an enzyme called ATP synthase. And as you might get from the name, ATP synthase synthesizes ATP, so it makes ATP. And what happens, those hydrogens rushing through, because they're going to go from where there's more to where there's less. So they're going to go through this transport protein, and that passage of those hydrogens activates this enzyme and you make your ATP. Then when the hydrogens get to the other side, remember what is making them move is that there's more hydrogens here than there is here. So we need to get rid of the hydrogens as they come out the other side, because if they reach equilibrium, then they're not going to come through anymore and you're not going to make any more ATP. So we get rid of those hydrogens by combining them with the oxygen we're breathing and making water. So that means there's always going to be more hydrogens here than here, and they're going to keep coming through, and we're going to keep making ATP. So the reason you're breathing that oxygen is to combine with these hydrogens coming through this enzyme membrane protein to make water, so that they're going to keep coming through and you're going to be able to keep making ATP. So that's the overall process of the electron transport chain. So what's fueling this pump is the energy from those high energy electrons in the NAD and FAD. That's pumping the hydrogen. They're coming back across, combining with oxygen to make water, but that's where we're making most of our ATP. So during the, let me go to the next slide, I think. And this process of the hydrogen coming through and making ATP is kind of on a molecular scale, kind of like water going through a dam, like at Roosevelt Dam, where they generate electricity. And the power from that water coming through is what generates the power. And it's kind of the same here. The hydrogen coming through is what generates the energy to make ATP. This little video, it probably will work on your computer if you want to watch it. It just kind of goes through the electron transport system. It doesn't really work great when I'm in record mode, so I'm going to skip it for now. This is just a summary of the 3 steps. And one thing I forgot to mention, but that's very important, is that in the electron transport chain, you make about 32 ATPs for every glucose that you're breaking down. So remember, in glycolysis, which happens in the cytoplasm, here's glycolysis, you made 2 ATPs and 2 NADHs. I'm going to change colors. And then in the citric acid cycle, you make some more NADHs and FADH and 2 more ATPs. And then all of those NADHs and FADH that you made in glycolysis and the citric acid cycle go to the electron transport chain, where they make a lot more ATP. So at the end of complete cell respiration, you had 2 ATP from glycolysis, 2 from the citric acid cycle, and about this, some variation, about 32 or 34 from the electron transport chain. So you get a total of about 36 or 38 ATPs for every molecule of glucose that you break down. So you can see, if you're not able to have oxygen and do the electron transport chain, you're only getting 4 ATPs from your glucose versus 36 or 38 with complete cell respiration. So that's why we need to do complete cell respiration to get enough ATPs to keep our cells alive. For some organisms that are able to survive in oxygen poor environments, which is anaerobic. So anaerobic again means without air, and it really means without oxygen. So if you were an organism that was living in an oxygen poor environment, you can't do complete cell respiration because that requires oxygen. You can do glycolysis, and you only get a little bit of ATP, but at least it's some ATP. So some organisms that can grow in anaerobic conditions do an alternate process called fermentation. And fermentation is a way, they still do glycolysis, they still make their pyruvate, but then that pyruvate can't go into the mitochondria for the citric acid and electron transport chain because they don't have oxygen. And so that pyruvate is instead converted to one of 2 things. One type of fermentation is alcohol fermentation, and that's done by yeast. And that pyruvate is converted to carbon dioxide and ethanol or alcohol. And so we use this process in making beer and wine, where we have organisms growing in kind of closed containers so that they don't have oxygen. And you put yeast in, and the yeast take the pyruvate after glycolysis, convert it to carbon dioxide and alcohol, and that's how the alcohol forms. We also use this in making bread. So when you have, you're making bread and you put yeast in, yeast is a living eukaryotic organism. It eats the flour and it produces carbon dioxide, which is a gas. Also produces the alcohol, but that gets the heat of the baking, breaks that down. But the carbon dioxide, the gas bubbles from the carbon dioxide is what makes the bread rise. And so that's what the function of the yeast there is. The other type of fermentation is lactic acid fermentation. And there, instead of converting the pyruvate to carbon dioxide and alcohol, you convert it to lactic acid. And that's carried out by bacteria, a number of bacteria, some fungi, and we sometimes do some limited lactic acid fermentation in our mussels when a mussel is being used so heavily that it kind of doesn't have enough oxygen to complete cell respiration. It will do some lactic acid fermentation as an alternate pathway. But other than that, we don't really do anaerobic fermentation particularly. This is just showing, so this is the glycolysis part where we're making pyruvate at the end. And what happens when we make pyruvate, we convert NAD to NADH. And remember we said that in cell respiration, that NADH goes to the electron transport chain, gives up those electrons, and is converted back to NAD. However, if you're not doing aerobic respiration, that NADH can't go to the electron transport chain, can't get regenerated. So you would eventually run out of NADH because you don't have any way of reconverting it to NAD. And then you wouldn't be able to keep doing glycolysis. And if that's your only way of making ATP, you're going to stop making ATP altogether. So with fermentation, if it's alcohol fermentation, when we're converting the pyruvate to alcohol and CO2, that regenerates the NAD, so you're remaking it. And likewise, lactic acid fermentation regenerates the NAD. So both of those processes enable organisms to keep doing glycolysis, and so even though they're only getting a small amount of ATP, they're still getting some ATP. And so they are able to keep doing that. So fermentation, again, is used in making beer, wine, some cheeses. The holes in Swiss cheese are from fermentation, carbon dioxide. Lactic acid fermentation is used in making yogurt. There's bacteria, like if you ever read the label, it says contains live cultures. That means it contains live bacteria that are eating the lactose in the milk and converting it to lactic acid by fermentation. And that's what changes the milk and makes it taste a little sour. It's that lactic acid that's forming. And it also is what kind of curdles it and gives it that different texture, is the milk, the lactic acid. So fermentation, you don't get nearly as much ATP from your glucose, but if it's the only way you can make ATP, it gives you a way of keeping glycolysis going to keep making some ATP. It's not enough, like we're not able to survive on fermentation alone, so we wouldn't be able to use that. That's why we can't survive without oxygen. So again, because you're only doing glycolysis with fermentation, you're only getting 2 ATPs for every glucose, compared to 36 or 38 when you're doing cell respiration. So you can see how much more efficient cell respiration is in producing energy for the cell. So again, with fermentation, you only get 2 ATPs, and that's actually from glycolysis. So again, we've already kind of gone through this, that with cell respiration, you get 36 to 38 ATPs, 2 from glycolysis, 2 from the citric acid cycle, 32 to 34 from the electron transport chain. In terms of our efficiency of getting the energy available from glucose out to make ATP, we're not totally efficient. We only get about 40% of the available energy out of a molecule of glucose, and the rest of that is given off as heat. So our body heat is the rest of the energy released from the glucose molecule when we break it down that we weren't able to convert into ATPs. And again, we've given the example of it doesn't allow me to go backwards. Through this process, we use the example of glucose as the source of making ATP. But remember, we can also break down fat. So if you haven't eaten for 3 or 4 hours, you're breaking down fat as your major source of energy. And when you break down fatty acids, you form acetyl-CoA, which is goes right into the citric acid cycle. And so you don't do the glycolysis part, but you do the citric acid cycle and the electron transport chain for making ATP from fat. Likewise, amino acids can be broken down to make ATP. We prefer not to use proteins as a source of energy, but we can. And they, depending on which amino acid, they enter into the pathway at different places.